island, occupying large fractions of Delta County, Alger County, and Schoolcraft County. And the east zone of the Hiawatha National Forest occupies large fractions of Mackinac County and Chippewa County, near landmarks in towns such as Brevoort Lake, East Lake, the towns of St. Ignace, Trout Lake, Rudyard, Moran, places like that. This is actually a zoomed in location of the same thing that I was just talking about. So I'll now move on to telling you about the surveys. And there's so much I could talk about, so little time. So again, I am just emphasizing two sites, and they both happen to be on the east zone of the National Forest. Just as much great stuff was encountered on the west zone. It just happens that I got the highest concentration of good photos at these two east zone sites. So the first one I'll talk about is a peatland complex in Chippewa County that I surveyed on July 15th. And this is aerial imagery or satellite imagery of that peatland complex. The actual survey area is that polygon with the cyan colored or light blue lines around it. And the reason I liked this site so much is because it was so heterogeneous. So you can kind of get a feel from that from the aerial imagery. This narrow strip of forest here is actually some upland woods. South of that in that lobe was some bog. North of that woods is a northern shrub thicket. West of there, it opened up into some fen or maybe bog. This big pond is a submergent marsh. West of there, there's like a marshy area with some cattail. And further to the west, there's a lot of fen. And in the far west, a little bit of poor conifer swamp, a little bit of black spruce swamp. So I'll share some of the kind of landscape shots highlighting those natural communities that I just mentioned. So this is a northern shrub thicket. I like to share photos like this just to emphasize some of the challenging natural communities that we have to walk through or get to walk through. Looks like in this photo, we've got maybe some Salix pyrifolia and Ilex verticillata and alnus incana. So kind of a nice diverse shrub swamp, not just dominated by tag alder. This is a picture of the submergent marsh that I mentioned. It looks like there's a beaver lodge out there, some blue joint Calamagrostis canadensis in the foreground. Looks like there's some emergent colonies of emergent plants out there, maybe some Chamaedaphne colliculata, leatherleaf, and some graminoids. The surface of this pond was dominated by Brasenia schrieberi. Those are all the lily pad looking plants, water shield. This looks like kind of a sedge meadow kind of an area, probably to the west of that marsh that I just mentioned, dominated by some sedges. It looks like maybe Carex, lasiocarpa, and then we can see a bit of tag alder mixed in and some black spruce there in the background. This is kind of more of that marshy sort of area that I mentioned. We see a lot of cattail. This is our native Typha latifolia, and there's some tamarack there, and looks like maybe some Eriophorum tenellum and northern blue flag, iris versicolor there in bloom. This is more of a fenny kind of an area, a fen west of that marsh that I mentioned. We see tamarack and maybe some Aronia prunifolia there in the central foreground, and then it looks like there's some shrubby cinquefoil, Dasyphora fruticosa there in the left side of the foreground. More of a fenny kind of an area with some dwarf birch or bog birch there, Betula pumila in the foreground. And now I'll share some close-ups of some of the cool plants that I encountered at this particular survey site. So this is marsh skullcap, Scutellaria galericulata, pretty common wetland plant. And if we zoom in here, we can see the calyx, which is said to be dish-shaped in this group of plants. And we can see this little knob-shaped structure on the calyx that's said to be shield-shaped. They call that a scutellum, characteristic of this genus Scutellaria. You can see some Chamaedaphne colliculata there as well, some leatherleaf. Moving on to the next plant. Here's uh, the one I mentioned dominating the submergent marsh, Brassinia schreiberi, the water shield. I think this is a cool plant because it grows in a cool habitat, soft water lakes and ponds, which tend to have a lot of other neat plants. Another nice one I encountered in this pond was Juncus pilocarpus. And it's also an interesting plant because of its phenology. This is a close-up of a flower. And they actually have two phases, these flowers do. They've got a male phase and a female phase. So the male phase is when the anthers are elevated above the stigma, as you can see here. That phase lasts about a day and then it sinks underwater and it emerges like the next day in the female phase when the stigmas are higher than the anthers. I didn't get a good photo of the female phase, but there's some nice examples on Michigan Flora Online. There were some nice orchids in this peatland. I saw some Malaxis unifolia, which is an adder's mouth in bloom. Didn't get a good photo, but I did get some good photos of these Platanthera species. We've got Platanthera clavellata on the left and Platanthera lacera on the right. And that Platanthera on the right, I just love that because it kind of looks like a little person. The fertile parts of the flower have these two kind of dots that look like eyes, and then we've got a couple of arms and legs and the main axis of the person's body there. Here's another shot of Platanthera clavellata. Like it's maybe growing with some Carex oligosperma. This is, what is it? Myanthemum trifoliata, or maybe it's trifolia, but in any case, this particular Mayflower, it's said to be not observed in fruit particularly often, so I thought it was kind of nice to get a photo with these developing fruits. This is a member of the sedge family. This is Eriophorum tenellum. So this is one of the cotton grasses, as you can see from the cottony fruiting head, which assists with wind dispersal of the fruits. I really love to observe Calla palustris, wild Calla. It's just so beautiful when it's in bloom. Here it is in early fruit. I also really like it because you can catch this plant in bloom or in fruit over such a long season. You can catch it flowering as early as May and fruiting into September. Homerum palustre, marsh cinquefoil, is always nice to observe. It's in the Rosaceae, as we can see from the leaves. Those leaves look a lot like Rosa palustris. This is a cinquefoil. Its flower looks a lot different from our cinquefoils of the genus Potentilla, so I guess it's no surprise that this one was split off and put into its own genus. We can also see the sedge Dulicium arundinaceum, three-way sedge here, with its characteristic three-ranked leaf arrangement. This is marsh St. John's wort, Triadinum fraseri. So this one's in the Hypericaceae. Michigan Flora's got it in its own genus, Triadinum. And I like this photo because it's said to be uncommon to observe this plant with its flowers open. But there's one with about 3 open flowers there. This is the bog aster, Oclamina nemoralis. I have a characteristic plant actually of poor fens. This is swamp candles, Lysomachia terrestris, with its beautiful inflorescence. Like Lysomachia is very distinct when it's in bloom. Before or after it's in bloom, it can be maybe a little harder to identify. Normally has opposite or whorled leaves. And a nice trick to use in the field, if it's a sunny day, you can rip off a leaf, hold that leaf up to the sun, and then the light coming through the leaf will reveal these purple dots all over the leaf blade. which will tell you that you do indeed have a Lysomachia. We've also got a Glyceria in the foreground. This is an important genus when it comes to wetland grasses. That looks like maybe Glyceria canadensis. Here's a nice sedge. I think it's a nice sedge, Carex mishoxii. This one kind of maybe could be confused with a member of Carex section Lupulini. It maybe kind of looks like Carex intumescens, but Carex mishoxii and its close relative C. folliculata have much longer, a much higher length to width ratio on their perigynia than would a member of Carex section Lupulini. This is another sedge. This one was actually the impetus for the survey. The purpose of this particular survey was to update an old record for Carex wigandii, and this one is special concern. I really like the story of this sedge because although it is a native element of the flora of Michigan, it was not detected in the state until the 1980s, and it's now known to be uncommon but widespread in the Upper Peninsula. So it's kind of cool that a native plant can lurk undetected for a century and a half or more. Still today, people are finding new state records, and then once they make their way into the flora, once botanists know to look out for them, sometimes they turn out to be more common than we realize. Okay, this is that same sedge, Carex wigandii. There on the left, we can see its affinity with Carex section stellulite. It's got those characteristic, they call it a clavate base of staminate scales on the terminal spike. More about that momentarily. And on the right, I'm trying to emphasize its wide leaf. Carex wigandii has a much wider leaf than its close relatives. And here's a picture emphasizing its clumping habit occurring there with some rhododendron, Greenlandicum, and some, that's probably viburnum lentigo. So that actually wraps up the first site. And with that, this is a nice segue into my first digression, if you will, because I want this to be not just beautiful plant pictures, but also practical information for your daily life when it comes to plant identification. So I do want to talk about identifying Carex, the sedges. Carex, I think, is a very challenging group. It's got so many species. We've got 184 in our flora within the state of Michigan. So it's by far our most species-rich genus. And this is true in much of the temperate world. And it's also challenging because it's got a peculiar flower morphology and inflorescence morphology. But it's a group of plants that botanists tend to really love because I think because they're challenging, because they're mostly native or even the non-native ones, they're not a problem. They tend to be pretty conservative. They occur in cool habitats. And it's so important to the wetland botanist because although we certainly have a lot of upland sedges, we seem to have more wetland sedges. And not only do we have a ton of wetland sedges, but we have a ton of common ones and even a lot that are frequently dominant. So it's really important to know your sedges, and there's different strategies for learning sedges. For example, you could kind of go off to Stalt. Oh, that's kind of got a particular look to it. That must be Carex lacustris. Nothing else looks like that. That kind of a thing. But you can only do that for so many species. What you really need to be able to do is work through the key. So we'll kind of talk about some tricks to help you do that. And let's begin with a broad introduction to Carex. We'll talk first about their inflorescences. So they do have unisexual flowers, unlike most other genera within the Cypripedium, within the sedge family. The sexes can be born on separate spikes in some species, as in Carex plantagenea here. This is a male spike. Some people call these spikelets. I learned off of Michigan Flora, so I follow their terminology and call them spikes. This is a female spike, as we can see from the white stigmas sticking out, as opposed to the yellow anthers sticking out of the male spike. Each spike is subtended by a bract, reduced leaf. Zooming in to talk in more detail about the flowers, the female flower is within a periginium. A periginium is a structure unique to Carex, and it is a highly modified bract. It's kind of shaped like a green flask or a bottle, and it's enclosing the entire pistil except for the stigmas, which need to protrude in order to capture pollen. The female flower of Carex is nothing but a pistil. There is no perianth. It's just a single pistil. And again, it is enclosed by that periginium. The male flower of Carex is nothing but 2 or 3 stamens. I forget if it's 2 or if it's 3, but that's all the flower is. It's just a very small number of stamens. Again, there is no perianth. That is, no sepals or petals. Here's a stigma protruding from a periginium. And each flower is subtended by a scale, a highly reduced leaf. So there's one subtending a female flower. There's a scale subtending a male flower. I already mentioned the bract. Here's a closer view of some perigynia. This is Carex vesicaria, a very important wetland sedge. There's one female spike, one spike period visible in this photo. Again, there's a periginium. Each periginium is subtended by a scale. The fruit in Carex is an achene. An achene is a dry indehiscent. That means it doesn't split open. One-seeded fruit that seems more like a seed and in fact functions like a seed. Carex tuckermanii is a pretty important wetland sedge. You can see the remnants of the periginium at the bottom of the achene, and you can see the style. It's really hard to get an achene out of a periginium with the style intact. This is a very nice photo. Carex can also have bisexual spikes. So a spike with both a staminate and a pistillate portion. As in Carex interior, there's the male portion. This is in section stellulity, just like Carex wigandia, which I shared earlier. So it's got the characteristic, what they call clavate base of staminate scales on the terminal spike. There's the female portion. And it's less evident in those species with bisexual spikes from other sections, but you can see it here in Carex trisperma. The female part is very obvious, perigynia with an achene within it. The male part is less obvious. Right now, there's just the filament, just the stalk of the anther visible, but that filament is subtended by a male scale there. That was a broad introduction to the reproductive morphology of Carex. I'm now going to share 3 particular tricks for working your way through the key. So we'll start with couplet 50 of the key to the sections. So a section is a taxonomic rank that is broader than species, but narrower than genus. So a lot of genera are split into sections, but there's really only one genus for which the sections are intensely emphasized on Michigan Flora, and that is Carex. It's such a big genus, really helpful to know the sections. Before you key out the species, you first have to key it out to the section. So couplet 50 reads, Bract of lowest pistillate spike, sheathless, at most with a thin scarious sheath 1 to 3 millimeters long, versus bract of lowest pistillate spike with a sheath about 4 millimeters or more long. So what is it talking about here? The two photos on the left represent sedges without much of a sheath on the lowest pistillate spike. The two on the right do have a substantial sheath. So let's zoom in on this, on Carex lamosa, fairly important wetland sedge on the left. Now, when I was first learning to key out Carex, I looked at the culm. The culm is the stem, and I thought the area above the bract of the lowest pistillate spike, this is the lowest pistillate spike, this is the bract. I thought the culm above that bract is very narrow relative to the culm below that bract. So to me, that meant the entire culm was sheathed below the bract. In other words, there was nothing but sheath. But it turns out what you actually need to look for is the node. So you can see the node is right here. A node is a part of a stem or a branch or a leaf shoots off from. And I kind of know just how to see them now, but I didn't realize back then. And usually it is pretty prominent just with the eye. But if you can't find the node, what you do is run your thumb and forefinger up and down the stem, and you'll be able to feel it. So if you look at that node, above that node, there's no sheath. So that's a sheathless bract. Whereas Carex vaginata has a node about here. The bract would be coming out about here. And so you can see there's a very substantial sheath there. Same with Carex plantagenea. The node would be about here. The bract, it's a reduced bract in this case, is here. So there's a long sheath. So that's couplet 50. The next trick I want to share is in regard to couplet 13. Styles 2 cleft, achenes 2-sided, versus styles 3 cleft, achenes 3-sided, or nearly terete, that means round. So on the left, we have Carex rosea. It's an upland sedge, actually, but I really like this photograph. They call this one twisty-styled wood sedge because that stigma is so curly. So it almost kind of looks like this is a 3-cleft style. It almost kind of looks like there's 3 stigmas, but what it is the stigma on the right is kind of curling around the back of the perigynium and back over. So this is a 2-cleft style, whereas Carex intumescens, very important wetland sedge, as you can see from the photo, has a 3-cleft style. Now, why is this couplet tricky? Because the style is an ephemeral part of the plant, doesn't last long, it just shrivels up and falls off after pollination. So during most of the sedge identification season, these aren't really available. But there's a lot of characters that are correlated with having either a 2-cleft or 3-cleft style. So if you know those, you don't have to belabor couplet 13. So what are those characters? The biggest thing is to look for whether the spikes are unisexual or bisexual. If they're unisexual spikes, it's normally a sedge with a 3-cleft style. So we've got a couple of important wetland sedges here. On the left, Carex lupulina, and in the middle, Carex camosa. They are each showing a male terminal spike and female, and approximately 4 to 5 female spikes below that male spike. So these have unisexual spikes, and they do indeed have 3-cleft styles. Very same with Carex ferrigula as the previous two. As opposed to species with bisexual spikes, those, I think always, not almost always, at least in Michigan, and everything I'm saying pertains to Michigan. I'm not sure how it would go elsewhere in the world. But in Michigan, sedges with bisexual spikes have 2-cleft styles. As we can see with Carex brunescens, fairly important wetland sedge, it does indeed have a bisexual spike. So the This whole thing would be a spike, and then this would be a little spike beneath it. But anyway, if we look at the terminal spike, we can see the very evident pergenia. And then lower down on the spike, we can still see the stamens with their anthers still intact. So that's a bisexual spike. So that species would have two cleft styles. Same with Carex rosea. Again, it's an upland sedge, but I really like this photograph. This is when it's in flower. It'd be very easy to tell it's got a two cleft style. I included this photo to show that it does have uh, stamens on the spike as well as pistillate flowers on the same spike. But if you saw it in fruit, it'd be a lot harder to tell. But there's other characters that I will go over that'll help you make this determination. But this one actually does have empty at this stage staminate scales. So you know that there were staminate flowers there at one point. So you want to look for whether the spikes are unisexual or bisexual to determine how many stigmas it has. There's some other helpful features. Sedges with two cleft styles generally have compact spikes, that is kind of short and fat. They're generally sessile, that is attached directly to the culm, not on a peduncle. The bracts tend to be small. The bracts subtending each spike tend to be small. The leaf bases tend to be green to brown, and that achenes are two-sided. In other words, lens-shaped, lentil-shaped, or lenticular. Sedges with three cleft styles generally have elongate spikes, which are often pedunculate, that is often on attached to the culm via a stalk called a peduncle. The bracts tend to be well developed and leafy. The leaf bases are often reddish purple, and the achenes are round to three-sided. That is, they're terete to trigonous. Now, there are two major exceptions, and the only way to deal with this is to just memorize them. Fortunately, these two sections are both pretty common sections without too many species to deal with, and most of the species within these sections are pretty common. So we've got section Phacocystis on the left here, represented by the very common wetland sedge Carex crenita. And we've got a fairly common wetland-ish sedge on the right there, nutty nibble Carex aurea. Now, looking at these two sedges, you would think that they have three cleft styles because they have unisexual spikes. There's a male terminal spike and then female spikes below. On especially Carex crenita, we can see the extremely leafy bracts. So we would think both of these have three cleft styles. They actually have two cleft styles in these two sections, so you just have to memorize these. That's trick number two. The final trick I'll share is in regard to couplet 23 of the key to the sections, which reads, staminate flowers at the base of some or all spikes, not at the apex. Note especially the terminal spike, versus staminate flowers at the apex of some or all spikes. Even when anthers have fallen, protruding filaments are usually visible. So on the left, we've got Carex normalis, pretty important wetland sedge, with staminate flowers at the base. On the right, we've got Carex cephaloidea. It's a little easier to tell with the one on the right. This is also an important wetland sedge because there's still anthers attached to some of the filaments. So we can see that those are at the top of the spikes. But how do you deal with it in the normal situation when the anthers, which are very ephemeral, are already gone? There's some clues that you can use to determine the relative position of the staminate flowers. The first is, just as the key says, look for filaments. So Carex trisperma, important wetland sedge on the left, we can see about 1 to 3 filaments per spike there. In the lower portion of the spike, there were formerly anthers attached to those. So that's telling us that the male flowers were at the bottom of the spike, as opposed to Carex sparganioides. It's maybe a little harder to tell here. I think these here must be It's hard to tell from a photo. It's easier in real life, but I think these must be filaments. There must have been anthers attached to those. This looks like a cluster, maybe of some staminate scales. So this would be one with the staminate flowers at the apex. So the key says to look for the filaments. Great trick. But the filaments too are ephemeral. Those two will fall off and they'll get crowded out by the developing perigynia. What I think works even better is looking for empty scales that were subtending a male flower, but the male flower is all gone. So we've already talked about it a little bit with Carex section stellulite, a member of which is Carex interior. So let's look at the photo on the right. There's a big, what they call, again, a clavate base, very prominent on the terminal spike, also evident on the lower spikes. All those empty scales were from male flowers. Here's what it looks like when they're at the terminus, at the apex of the spike. So these are some empty male scales on Carex rosea. And we can also kind of see it on some of these Carex sparganioides spikes. So those are some tips and tricks for identifying Carex. And next I'll move to the second of the two sites that I want to share with you. And this was a fen complex in Mackinac County that I surveyed on July 16th. And the cyan or light blue colored lines, those were my GPS tracks. So that's the area that I walked within this wider landscape. And I'll just get those out of the way. And you can see some kind of open, marly areas of the fen within this wider conifer-dominated landscape. And I'll share some of the interesting plants that I observed in this fen complex. This is dwarf mistletoe, Archeothobium pusillum, which parasitizes spruce trees, typically black spruce. So in that sense, it's a wetland plant. There's some more. That is a flowering plant. It's non-photosynthetic. It doesn't look much like a plant. Here's grass pink, Calopogon tuberosus. So this is one of our 4 common peatland orchids that have pink flowers. And this one is kind of known for being non-resupinate. So normally, orchid flowers resupinate, so the bottom flips to the top during development. And the fertile parts are normally at the top. In Calopogon tuberosus, they're at the bottom. A couple more photos of grass pink here. Sarracenia purpurea, pitcher plant, needs no introduction to members of the Michigan Wetlands Association. This particular Spiranthes, this is another orchid. This particular species of ladies' tresses, I keyed out to Spiranthes romanziana, or romanziana alpha. Sorry, however you say that specific epithet. It's a challenging key to work through the species of Spiranthes, and actually the taxonomy has been updated more recently than the key, so who the heck knows, really, but a very lovely ladies' tresses in any case. So here we can see kind of the open marl-rich area of the fen, and these are, again, in a landscape of conifer forest. And there were some very physiognomically interesting conifers on the ecotone between the woods and the open fen, in particular, white pine. I've never quite seen this sort of windswept looking growth form of white pine as I did on the edges of this fen. This is Lakeside Daisy, Tetranuris herbacea. Not a great photo, sorry, but I felt like I had to include it because this one is state endangered and federal threatened. So this is an extremely rare plant from a global perspective. It's past fruiting here right now, so it's already dispersed. So, but all these plants here are lakeside daisy, and it's co-occurring with this aster-looking plant that's actually Solidago tarmacoides. I think they call that one upland white goldenrod. Now, this is a nice open section of the fen. Very wet there. As I mentioned, it opens up or it grades into conifer forest. So here's kind of a sparse swamp of stunted white cedar. There were actually 5 total rare plants in this fen. So this is the second one I'll share with you, and this is uh, Carex serpoides, or is it Carex serpoidea? In any case, this is a special concern sedge. Here's some more of it. This is another special concern plant here. I'm talking about the yellowish colored basal rosettes, and those are bladderwort, Pinguicula vulgaris. This one is state threatened, very rare plant, known from just 2 counties of the Upper Peninsula, and this is Erigeron hyssopifolius, daisy-leaved fleabane. Looks like it's in fruit here. Not a great shot from the perspective of getting plants into focus, but I just kind of wanted to talk about these hummocks. This fen was peppered with these really diverse hummocks, and a lot of them supported up to 3 rare plant species. They would have Pinguicula vulgaris, as this one does, Carex serpoidea, and also Erigeron hyssopifolius, which it's kind of hard to tell from this photo if those white flowered plants are that species or not. But in any case, some of them did have all 3 of those species. And we can also see some other conservative fen plants here. This looks like Triantha glutinosa, and this looks like Parnassia glauca. Very nice hummocks. This one is the federal and state threatened Houghton's goldenrod, Solidago houghtoniana. So just a really cool site. Okay, so I do have time to take one more digression about plant identification, and this is in regard to the rushes, to the genus Juncus. So unlike sedges, which have no perianth, um, Juncus does have a true perianth of 6 tepals, or if you like, 3 sepals and 3 petals, as we can see on path rush here. Juncus tenuis, and also on this other rush, Juncus stygius. Now, they're not all this showy, they don't all have this beautiful of a perianth, but they do all have a perianth. The fruit of Juncus is a many-seeded capsule. So a capsule is a fruit that dries out and dehisces or splits open across multiple sutures or multiple kind of lines of kind of splitting lines, as you can see on this photo of a Juncus fasciae capsule on the right. And on the left, we've got about 5 capsules of J. dichotomous with many seeds spilling out on that white background. The inflorescence in Juncus is either highly branched, as in Juncus biflorus here on the left, or it's much condensed into heads, as in Juncus torii here on the right. Usually these are wetland plants, as we can see from the photos here. So Juncus is a very important genus to the wetland botanist as well. But I do think that they're pretty tricky to key out. So I'm about to share with you several tips and tricks for keying out Juncus. It's a lot less species rich than Carex. We've got maybe 25 or so species in our flora, and a lot of those are actually pretty uncommon. But there's a handful of very common species to know, and then there's another handful of species that are pretty common, but only in the correct habitat, so you don't encounter them as frequently, so harder to get a gestalt-based, harder to get the search image, as they call it. And so You often have to key them out, and it's important to be able to work through that key efficiently. So let's go over some tips and tricks. First is couplet number one. Inflorescence apparently lateral versus inflorescence terminal. I've got those ellipses there because there's more to the couplet, but this is the part of the couplet I'm emphasizing. So what it means is, does the inflorescence appear to be just coming out of the side of the culm, the side of the stem, as in Juncus effusus, very important wetland rush, one of our most common rushes overall. The inflorescence appears lateral because the top of the culm is actually where the inflorescence is coming out. That's actually technically the terminus of the culm. But above the culm, it looks like the culm just continues. That's actually a bract. That's a bract subtending the inflorescence, but instead of kind of coming out perpendicular or at an angle relative to the culm, it's just appears to be, it's parallel with it. It's just going straight up. It appears to just be a continuation of the culm. So it's apparently lateral, as opposed to most other Juncus species in our flora, the inflorescence of which is terminal. As in J. canadensis, another one of our very most important wetland rushes. The next tip and trick is in regard to couplet 6, leaf blades flat, involute, or terete, but without hard cross partition. So that means leaf blades flat or kind of curled up and channeled, involute, or terete rounded, but without hard cross partitions, as opposed to leaf blades more or less terete, maybe flat and pressing, hollow with hard cross partitions at regular intervals. Now, this one on the right, presumably that's a pressed and dried specimen. And so the hard cross partitions are very evident just with the eye. But normally the best way to determine if there are hard cross partitions, and it actually mentions this right in the key, but the best thing to do is to get it against a flat, hard surface, like a field notebook, and run your fingernail, your thumbnail, along all the way up the leaf. And if you feel resistance, if you feel little bumps at regular intervals all the way up the leaf, those are the hard cross partitions. The next tip and trick is about couplet 22, the first half of which is seeds with a long or short pale appendage or tail formed by prolongation of the seed coat at both ends, as opposed to seeds without pale tails, either blunt or with small dark points at one or both ends. So when I was first learning to key out plants, I found it very intimidating when there was any sort of dissection involved, if you will, like any sort of a structure that was hidden in any way. So I found this a bit baffling at first, but it's actually very easy. So all you have to do is get the seeds out. And the nice thing is that for most rushes, ripe capsules, ripe fruits are available throughout much of the growing season. So usually, anytime you need to determine this couplet, the material is available. And so all you do is you pull out a capsule with your thumb and fingernail, with your thumbnail and your middle fingernail. And actually, it's very easy. You just pinch the middle of the capsule and you pinch it perpendicular to the length of the capsule. So kind of like you're making a cross section, and you just pinch right there. It's important in botany to keep just a little bit of a fingernail so you can do things like this. And you just pull the top of the capsule off, sprinkle the seeds on your hand, and then you just look at them. And you can usually tell pretty easily just with the naked eye if they have these two long appendages of a pale color, as we can see in the zoomed in Juncus fascii. And it's also very characteristic of J. canadensis and some other rush species, as opposed to seeds without pale tails, as we see in Juncus articulatus. They do have kind of 2 points at the end of the seed, but they're blunt and they're dark in color. The last Juncus tip and trick is about couplet 25, stamens 3, opposite the sepals, or stamen 6 opposite the petals and sepals. And this is kind of like our Carex discussion. This is tricky because the anthers are ephemeral, they fall off. However, the filaments in juncus, the filaments of the stamens, tend to persist for a long time, even deep into the fruiting period. So usually, anytime you need to work through this couplet, usually the right material is available to you. It's very evident when they're in flower, as we can see with Juncus canadensis on the left. It's actually a little bit hard to tell from this photo, but you can imagine just with those intact anthers that it'd be a relatively easy determination in the field. And we can tell quite obviously that there's more than 3 stamens on each of these flowers of Juncus compressus here. So let's go into a little bit more detail about this one. Because there's that thing about, are they opposite the sepals? Or are they opposite both the petals and the sepals? What you're looking for is one or both of the following things. If you find filaments on 2 consecutive tepals, or if you find at least 4 filaments total, it's a species with 6 stamens. So if there's only 3 stamens, they're going to each be opposite from a sepal. They're not going to be opposite from a petal. And the sepals and the petals alternate. Or if you like, just call them multiples. So if you see a stamen on 2 consecutive tepals, you know right away that there's one of them is opposite a petal and one of them is opposite a sepal. Therefore, it has 6 total stamens. Or if you're having a hard time seeing the 5th and 6th stamen, but if you just very quickly see there's 4 stamens or more, then you know it's not just 3 stamens, it's going to be 6 total stamens, as in Juncus stygii there. So those are the 2 sites I wanted to share with you. Those are the 2 groups of plants I wanted to talk more about plant ID about. I do just want to take a moment to thank all of the photographers on Michigan Flora whose photographs I borrowed. I did get permission from all of those. I know how hard it is to take good pictures of plants, and I really appreciate your efforts. Also want to thank my colleagues at MNFI who I worked on this project with this year. Wasn't able to include everybody from previous years. Tony Resnicek helped with some specimen determinations. And of course, Michigan Flora was frequently consulted. I also want to thank our partners at the sponsoring agency at the Hiawatha National Forest, Paul Thompson and Justin Bourneville. And I especially want to thank Chelsea Atterbury, Ted Roper, and Robert Lieberman for joining me in the field and providing critical knowledge. With that, MSU is an equal opportunity and affirmative action employer. And with that, I'll take any questions.
Well, thank you so much, Scott. These photos are just absolutely gorgeous. And the detail that you put into this presentation, we really appreciate it. I have probably 6 to 10 pages of notes. So thank you. We'll go through the questions. I do see one that just came in. This one's from an Amber Hubbard. It says, do you have any plans to survey Round Island just south of Mackinac Island?
Wow. Well, first of all, Brianna, thank you very much for those kind words about my presentation. I really appreciate that. Amber, no, I don't have any plans to visit that particular island, but I sure would love to. I haven't been to Mackinac since maybe like 2003. I'd love to get back there. Would love to go to Bablo or Bois Blanc, as they call it. And I would really love to get to Round Island, but no immediate plans to do so.
I guess I don't see any other questions, but I had one. What locations are you looking to study this year, if you are?
Well, let's see here. I haven't heard from the East Zone botanist, but the West Zone botanist got in touch with me. Gosh, I can't remember where all he wanted to survey. I know there was a really cool fan in Schoolcraft County that he wanted to survey. I'd really like to get back to Grand Island and look for Crataegus douglasii, Douglas's hawthorn. There's a lot of cool fens in like Schoolcraft County and um, maybe back into like the Jack Pine Plains of Delta County. So not too sure yet.
Okay. It says, do you have, what are your favorite resources for sedge or graminoid ID? You probably have a few.
Yeah, well, it's a great question. Graminoid identification is just so important. Let's see here. Well, I mean, more and more people are using iNaturalist, and it's a great tool. So I think iNaturalist is a really great way to build up your gestalt-based identification to kind of quickly learn a lot of groups of plants. I already knew my vascular plants before I kind of got really into iNaturalist, but it's just been so terrific for learning about groups of organisms I don't know, like birds and herps and non-vascular plants. Is it perfect? No, of course not. But as I say, to quickly learn a whole lot of things in a non-technical fashion, it's just terrific. So with the graminoids, it's still got a long way to go. AI-based identification within iNaturalist, but it can at this point, if you take a good photo, get you pretty quickly and accurately to genus, and then you have to key it out from there. So iNaturalist is a great one. Michigan Flora, I really can't speak highly enough of. Just a critical tool to every botanist in Michigan. We're so fortunate to have it, and most states don't have something like this. So using Michigan Flora, really kind of getting used to the keys on Michigan Flora, really reading it carefully and looking at the introductory essays to the families, the genera, and reading the species profiles, as well as the information in the keys. Now, they don't have time to explain everything. There's a lot of weird tips and tricks you have to learn, kind of like what I was getting at in the presentation. I already went over grasses and rushes, excuse me, sedges and rushes. Grasses too are really tricky. As they say on Michigan Flora, you really have to know what you're looking at. So just really learning the morphology of grass inflorescence and flower is really important. So yeah, I just really stress Michigan Flora. Um, Joining societies like the Michigan Wetland Association and Michigan Botanical Society and getting access to their field trips. Most people have heard of the annual foray for Michigan Botanical Society. Most of the chapters also have regular Saturday field trips. These are freely available to you with an affordable membership. Going out with botanists like that is such a great way to learn your graminoids. So many great websites nowadays. And I'll mention a few books. Paul Rock's got some great uh, sedge books out from an Indiana perspective. It's very relevant to the Michigan botanist too. Really awesome book on the oval sedges was just published out of Illinois. There's a great grass guide and a great sedge guide uh, from Wisconsin. So those are a few ideas.
Great. I tried to, in the comments, the question and answer section, I did try to type up some of your responses. So another question, it's pretty related to that, is what are some resources for getting better at using dichotomous keys? So probably what you've kind of summarized, Michigan Flora and using iNaturalist and other, sedge books, but I'll let you.
Well, oh yes, thank you, Brianna. So with dichotomous keys, you really kind of have to learn the um, I'll say eccentricities of each author. Just different authors have different ways of explaining things. So, like if you're using, for example, Barnes and Wagner's Trees of Michigan, or if you're using Michigan Flora, just each of us has our own kind of internal dictionary, right? So we each just kind of have our slightly own different definitions of words. So just kind of learning what they mean. Like a great example would be what I mentioned with kind of the basic unit of the sedge inflorescence. Is it a spike or is it a spikelet? Things like that. But the really big thing when it comes to dichotomous keys is all the terminology. It can be very cumbersome to the beginner. So just learning that terminology using the glossary, and practice, practice, practice. It's really great when you can get a botanist with more experience to work through it with you. Visiting an herbarium is a really good strategy, particularly a well stocked herbarium with a lot of specimens that you can compare your determination against to see if you were right.
Okay, thank you for the answer for that. That was very helpful. I think there is a couple books out there, right, for plant terminology, right? I think put in a link for one of them that I've used. So maybe following this presentation, we can pull together some of your botanical or field guides or any kind of key links that you would recommend, and I can help you pull that together if you'd like.
Yeah, we can certainly do that. The best known book for what you mentioned is Harris and Harris, Plant Terminology. Great book.
Okay, I think that's the one I put. But here is a question. The couplet comparisons were very helpful. Can you make your slides available? So it's up to you.
Yes, I'm very happy to do that. And thank you for that comment, by the way. I appreciate that.
Okay, well, I think it's just past 4, so we really appreciate you, Scott, for putting this together. So much information, beautiful photos, and we really appreciate everything. So, and thank you everyone who attended. We had about 70 people that joined on today, so that's pretty exciting. So,
Well, thank you so much. I really appreciate it.
okay. All right. Thank you, everyone. Bye-bye.
Bye.